Okay, back again once the camera seems to get in focus. So we're back to Biology 160. We're still working through worksheet number 6. I'm on page 26, number 7, doing these organelles, and they're listed in order, and that's the way that I'll go through them. So the nucleus, our first organelle, is inside all eukaryotic cells. So if this is our cell, inside it is typically a rather large organelle. We'll just call it the nucleus. And your texts have lots of pictures of nuclei, so you can get an idea of what they look like. But their main claim to fame, if you will, there's a couple of things. The main one is that the nucleus contains the chromosomes. Contains chromosomes. And the chromosomes are essentially the DNA. Those are your genes. That's the heritable material. All of that is inside the nucleus. So to draw another picture here, just kind of make sure we're all clear about this. Here is the nucleus inside, and this is the DNA. Your genes, all of your chromosomes that we'll talk about. It's a double helix sort of affair, and you have in humans you have 46 of those chromosomes. That's in the nucleus. Okay, the nuclear envelope, let's do like this. Here is another cell. I'll make this one fairly big, and I'll make the nucleus fairly big also. What I'm getting at is that the nucleus has a double membrane around it. So it's a double, each membrane consists of a phospholipid bilayer. So there's one phospholipid bilayer, and here is another phospholipid bilayer. And actually between those two there are pores. So if I redraw this, so this, let's label this as our double nuclear membrane. I don't know if it's all that important that you remember that it has two phospholipid membranes around it or not. But anyway, let's draw that nucleus again. There's a cell, and this time I'll draw the pores between it. So here's a piece of this phospholipid bilayer, piece of the nuclear envelope rather, and then there's another piece that goes like this. And there are lots of these pieces, but what I'm trying to show are the gaps between them. And so this is the nucleus. I'll just write nuke. And these are the nuclear pores. And what that allows for is, what we'll see later, is that DNA gets converted or is used to make RNA, and then the RNA can leave the nucleus through those pores and go out into the cytoplasm. RNA, let's say now, in cytoplasm. Because all RNA is made in the nucleus. Let's say using a DNA template or instruction sheet. We haven't talked about that yet, so don't worry about it. But we will. And if you remember that the nucleus contains the DNA in the nucleus, DNA doesn't get converted to RNA. DNA stays DNA, but DNA is used to make a new molecule of RNA. And RNA always gets made in the nucleus, and then it's able to get out through these pores. So that's enough of the nuclear envelope, I think. Remember that each one of these black lines represents a phospholipid bilayer. So it's two of those bilayers together. And when the cell divides, we'll see that this nuclear envelope breaks down, disappears, at least under the microscope, so that the DNA can get replicated and the cell can then divide in half, and then the nuclear envelope reforms. It's kind of an interesting process. But at any rate, I think that's enough of the nuclear envelope. The nucleolus, which sounds like nucleus but is not. So this is the nucleolus. The next thing I want to talk about. The nucleolus is a region within the nucleus. It doesn't have a membrane going around. So this is our cell. And here is the nucleus. So we'll just label this cell membrane, cell memb. And we'll label this the nuclear envelope. Okay, and the nucleolus is inside the nucleus. It's sort of a poorly defined area. It may stain a little darker, but I'll just represent this area within the nucleus as the nucleolus. And there's no membrane going around it. What you need to remember about the nucleolus is that this is the site where ribosome subunits are built or assembled. So we're putting the cart in front of the horse a little bit when we're talking about ribosomes, but we will get to them shortly. It's 3 or 4 down on the second one down on the list after nucleolus. So let's say that this is where ribosome subunits are assembled. So we're going to come back and talk about ribosome subunits. There are 2 subunits. There's a large and a small. And they're both built in the nucleolus. Abbreviate large and small let's spell it out, ribosomal subunits. So, there's the nucleolus. It's inside the nucleus. And it's where the ribosome subunits are put together, and then they will leave, those subunits will then leave the nucleus for the cytoplasm. So let's say that this is a small subunit and this is a larger subunit, and after they are built inside the nucleolus, they are moved out into the cytoplasm. So we have the small subunit of a ribosome, and we have the large, I'll abbreviate large, subunit of ribosomes. So then when we talk about ribosomes, you've already been introduced to at least the subunits and where they're made. Okay, the next organelle, or the next structure that I want to talk about, are chromatin strands. Okay, so chromatin strands. chromatin, that's an S, believe it or not. Chromatin strands, it's basically DNA. So chromatin, the name is, I guess, the historical roots come from the fact that you could see it. And it sort of changed color and became visible at different stages of the life cycle. So what chromatin strands are is DNA with associated protein. Associated protein. So this might be what it sort of looks like. And there are different pictures we can look at in just a moment. So if this is a piece of, let's say, double-stranded DNA, we'll look at the structure of DNA later in the course, but let's just call or label this double-stranded. This is a double helix DNA. And there are all kinds of proteins that are associated with it. And when you're looking at a cell underneath the microscope, you can add different stains to it that will bind to the protein and the DNA, and then it'll make it easier to see. So the green dots are proteins that are bound to the DNA. And that makes chromatin. And that's all there is to chromatin. It's DNA plus protein. And I had a picture. I don't remember if I can get to it, where that was, in which book I was in. But what you ought to look at, if I get a picture here, I may put it back on. What you should look at are, let's say, see Google Images for, let's say, chromatin, and the proteins are histones. So if you Google images for chromatin and histones, you should see some of these proteins. So the histones are the protein. And they wrap around the DNA more or less like this. So we'll stop here for the moment. I'll come back though shortly and with a picture of chromatin and histones, and then we'll finish that up and then move on to ribosomes.